To ensure the survival of their species, all organisms must reproduce. In the animal kingdom, this usually begins with the search for a suitable mate. From peacocks to humans, animals display their biological urges in many interesting ways.
Courtship behaviors have evolved because that's a way of convincing the partner. In most cases, it's the female that's being convinced that the male is the best possible male available. And in the case of some birds, that means the males with the brightest colors or the most beautiful songs. Of course, the same thing happens in human courtship behavior as well.
While it takes many forms, sexual reproduction has but one goal: to unite egg and sperm in the production of a new life. But how that fertilized egg develops the complex structures of a living organism remains one of biology's most intriguing mysteries. The residents of the animal world reproduce in many strange and interesting ways, but the process usually involves the union of gametes, ovum and sperm, in a fertilized egg, which then develops into the budding life we call an embryo. Dr. Marianne Bronner-Fraser and Dr. Scott Fraser are a husband and wife team who explore the fascinating world of embryonic development.
Each of our research involves how cells pattern, how cells migrate to the right place, recognize they're in the right place, and decide which cell types to make.
The Frasers' research depends on their knowledge of different reproductive strategies. While most animals combine the female egg and male sperm in sexual reproduction, some organisms can produce their offspring asexually, without a mate.
Asexual reproduction is reproduction that takes place without any kind of sexual intercourse. It's very simple, and it's not as common as sexual reproduction. One example of a really fascinating organism is one called Hydra. It reproduces by making a little bud off the side of its body structure, and that bud is like a miniature version of itself, which then grows and forms a head and a tail.
Asexual reproduction allows an isolated organism to make an exact genetic copy of itself. Using this strategy, simple organisms can multiply in stable environments where diversity is not crucial to survival. Most organisms, however, reproduce sexually by combining gametes from two parents. The new gene combinations provide these organisms with a key advantage in the struggle to survive.
You increase the genetic diversity because you have two organisms, two parent organisms, contributing genes to the offspring organism.
For all sexual species, the development of life begins in the fertilized egg, called a zygote. How this single cell gives rise to an entire organism has been a source of constant fascination to the Frasers.
The central question that all developmental biologists are interested in is really posed by that single fertilized egg. And that is, how do you get a complex organism from one cell? So the first step in getting that complex organism is for the cell to undergo mitosis. It divides to form 2 cells, and then 4 cells, and then 8 cells, and so on. And that process of dividing the single cell into many cells is referred to as cleavage.
While cleavage reveals the first signs of an emerging life, this mass of dividing cells is far from a complete organism. We see the first visible signs of the transformation to come in the migration of cells known as gastrulation.
Gastrulation is a stage where the embryo starts to undergo large movements in cells. This process of gastrulation takes your simple cleavage embryo and makes it a much more complicated and layered embryo.
These primitive cell layers will eventually give rise to the different tissues and organs of the body. Using special imaging techniques, the Frasers can document the embryo's progress as it determines each cell's destiny.
We inject a dye into a single cell, and the dye is a very large molecule, so the only way that it's passed on to other cells is by cell division or mitosis. So by looking to see what cells are labeled with this dye, we know that those cells were all related to the original cell that we labeled.
One of the advantages of the imaging approach we use is that we can follow an embryo from its earlier stages when it's just undergoing process of cleavage where it appears that there aren't any unique identities to the cells, through the process of determination where those cells are told to become different from one another. Okay, so
that's junk. That's on the outside.
Junk down there.
That's on the outside.
With their general future determined, the cells of the developing embryo begin to differentiate or specialize into distinct tissues.
Cell differentiation is the point at which a cell becomes distinguishable as a particular cell type. And there are many ways that cells become different. For example, in the case of a nerve cell, the cell has a long axonal process, and it's very clear and easy to recognize. You can clearly see when a cell has become differentiated because it is so different than the cell that it came from.
For these differentiated cells to form distinct tissues and organs, many events must occur simultaneously in a process called morphogenesis. Cells move, grow, and divide. Some even die to chisel out forms and shapes. Perhaps most critical, however, is the ability of cells to interact with each other through a process called induction.
Well, induction is a process by which one group of cells becomes different by contact with another group of cells. You can find this in the induction of the nervous system, or the underlying cells that will go on to make the backbone induce the cells above them to make the nervous system.
So an inductive interaction is actually a conversation between two cell types. One cell type that's giving some information and one cell type that's receiving that information. And without this kind of inductive interaction, embryonic development would never take place.
Through their groundbreaking research, the Frasers hope to discover more about the complex processes that build and shape living things. But these amazing transformations all depend on a single critical event: fertilization. Bringing a new human being into the world is a complex undertaking. All parts of the reproductive system must work as designed. But for Leslie Monroe and her husband Kevin Bankston, attempts to have a child had been met with loss and disappointment.
I think when we realized that having a natural pregnancy, that wasn't something that was really best for Leslie to go through, it was depressing. It was kind of something we really didn't want to accept.
We knew that we wanted to have a baby eventually. I'm not as young as I was, and just that if it's going to happen, let's try to make it happen now. Let's do what we can.
Okay, hi Lena.
Leslie and Kevin sought help from Dr. David Diaz, a specialist in fertility medicine.
In our practice, we tend to find the male and the female factor cause of infertility almost on an equal basis, so that we might say that in 40% of couples, we find a female factor, in another 40% a male factor, and in the remaining 20, there's a combination of both.
Male factors in fertility usually begin with the sperm.
We usually encounter problems with low numbers of sperm along with low motility of the sperm cells. And we also are focusing more and more on the morphology, which is the structure of the sperm cell.
Researchers often trace these problems to the male's primary organs for reproduction, the testes.
The testes, which is the male gonad, is the source of two important elements, that is the sperm as well as testosterone, which is the predominant male hormone. The majority of the bulk of the testes is composed of the seminiferous tubules that contain the sperm-generating cells. Now, interspersed in between the seminiferous tubules are the Leydig cells. These are the cells that are responsible for testosterone productions. So as the testes receive the signal from the pituitary gland, sperm cells are developed and matured in the testes initially. And this happens about every 72 to 74 days. From that point on, there is another period of maturation in another small gland called the epididymis.
Because sperm production is continuous, the epididymis contains sperm cells in various stages of development. They remain there until expelled from the body during ejaculation. At that time, the sperm cells mix with secretions from different glands. For example, the seminal vesicles provide volume and nourishment, while the prostate gland is responsible for neutralizing acid.
The vagina is very acidic, and in order to survive, the sperm must be packaged in their own environment.
Once sperm enters the vagina, the cells begin their long journey to seek out a waiting egg, first through the uterus, and eventually in the oviducts, or fallopian tubes.
The sperm are propelled upward to reach the egg by several different mechanisms. Number one is the motility of the sperm cells themselves. It's also felt that contractions of the uterine muscle related to orgasm also may play a role in propelling the sperm quickly upward toward the fallopian tubes. Many sperm are approaching from different angles. However, only one will ultimately be successful in penetrating through all of the membranes.
For fertilization to occur, however, an egg must be released by the female's primary reproductive organs, the ovaries. The ovaries produce their egg cells and reproductive hormones in a repeating cycle.
The release of a female egg occurs as part of a repeating sequence of events known as the menstrual cycle. First, the pituitary gland releases molecules of FSH, or follicle-stimulating hormone. In the ovaries, follicles are hollow balls of cells which contain a single, immature egg called an oocyte. FSH stimulates these tiny follicles to produce the hormone estrogen. This causes the oocyte to mature and the follicle to swell with fluid. Estrogen released in the blood also stimulates the growth of the uterine lining, the endometrium, by increasing cell divisions. Just before the middle of the cycle, the pituitary gland releases a second hormone called LH, or luteinizing hormone. The final spurt of growth that results ruptures the follicle. This releases an oocyte into the fallopian tube in an event we call ovulation.
The woman never produces new egg cells. The same egg cells that she was conceived with are the ones that she carries all her life. Most women will typically ovulate about 400 times in their entire life, on the average of every 28 to 30 days.
After ovulation, the follicle cells that remain in the ovary form a structure called the corpus luteum. This yellow tissue continues to produce estrogen and a second hormone called progesterone. In the uterus, progesterone prepares for the arrival of a future embryo by quieting normal contractions and stimulating endometrial cells to secrete important nutrients. If the egg is not fertilized, hormone levels drop and the endometrium breaks down. The uterus releases the resulting blood and tissue in a menstrual flow, after which the cycle begins again.
If an egg is fertilized, the future embryo will embed itself in the endometrium and emit a hormone that suspends the menstrual cycle. But if anything goes wrong with the cycle or the organs that carry it out, infertility can result.
I've had 2 ectopic pregnancies, tubal pregnancies, so with each tubal pregnancy, your risks increase. So after having 2, the chances were very, very great that I would have another one.
In Leslie's case, the ectopic or out-of-place pregnancy implanted in the fallopian tube with harmful results.
Based on what we know about ectopic pregnancies, we know that the fallopian tubes in those cases were probably damaged.
With this in mind, Dr. Diaz recommended that Kevin and Leslie try in vitro fertilization in the lab.
Like to sit home as the ants up in?
After a period of injections to stimulate egg production, Dr. Diaz harvested a number of eggs from Leslie's ovaries.
The needle will pass through the top of the screen.
We can scan the ovaries very accurately and pass a fine needle into each follicle to withdraw the fluid and the egg.
Inject one.
This is then transferred into a test tube, which then goes into our laboratory.
Rinse from R1.
Using Kevin's sperm, doctors fertilized Leslie's eggs in the lab, then placed them in her uterus 2 days later. But because in vitro fertilization offers a success rate less than 35%, there was still a time of uncertainty for the prospective parents.
But then actually...
Following the retrieval, waiting 2 weeks to find out if it took was a long 2 weeks.
This is the cutest, but that's too big.
When I found out I was pregnant, it was over the telephone. The nurse practitioner, Joan, who I dealt with most of the time, said, "You're gonna have a baby," and I don't know, I cried and couldn't believe it and didn't think I'd hear that, and I couldn't talk. I had to hand the phone to Kevin.
Helping couples like Leslie and Kevin build a family is the best part of the job for Dr. Diaz.
From both ovaries, so we are finished from this part.
We have an annual celebration for these patients, and they come to our program, and we have a party and a celebration, and we get to see some of the kids who are now 6 years old, and to watch them grow up is also really a special feeling for them and for us.
Conception marks the beginning of great change for the mother and developing child. In preparation, many prospective mothers make an effort to alter their lifestyle.
I used to smoke, and when I got pregnant, I quit smoking, and so I started putting on some pounds, and I decided I needed to exercise.
It gives me a boost that I really need for the day. This is good. I don't move real well, but it's fun.
Fingers, stretch them out. Keeping your rear ends tight. If you have strong legs and you have some stamina, you're going to be able to push that baby out a little bit easier.
During their pregnancy, most expectant moms work with an obstetrician like Dr. Peggy Grau to monitor the developing life inside them.
This is the front side of him. His shoulder's out here. You can see the heart beating.
A human pregnancy lasts 40 weeks, typically from the last menstrual period, or 38 weeks of embryonic and fetal development. It has been traditionally divided into thirds. The first trimester, which used to be the first third, when now has been reduced to the first 9 to 10 weeks, is the period of embryogenesis, or organization of the embryo from a small cell mass into a recognizable human fetus.
This miracle of organization begins as the future embryo, a ball of cells called a blastocyst, implants itself into the endometrium and begins to form the placenta. At the same time, through processes such as cell migration and differentiation, the embryo continues to develop with amazing speed.
By 14 days after conception, the embryo has organized itself into the different germ cell layers and is beginning to differentiate into different organ systems. One of the first ones that we're aware of is the neural tube, which begins development at 24 days of fetal life and is finished by 30 days. Very often, a malformation occurring in this stage will have already taken place before the mother is aware that she's even pregnant.
Although only 9 to 10 weeks long, more developmental problems occur during the first trimester than any other stage of pregnancy. If the defects are serious, the embryo could be lost.
The most common event to look for in the early part of pregnancy is pregnancy failure. It's estimated that somewhere between 25% and 40% of all human pregnancies fail.
During the second trimester, organ systems increase in mass and continue their development. Major changes occur in the nervous and reproductive systems.
At this stage, the external genitalia begin to develop. The eye is almost completely developed by the beginning of the second trimester. By the end of the second trimester, 24 to 25 weeks of development, the eyelids are open. The last trimester continues with rapid cell growth and increase in weight in the fetus, and is characterized primarily by continued central nervous system and pulmonary development.
During the second and third trimesters, Dr. Grau uses harmless sound waves called ultrasound to monitor the development of the fetus.
You can see the baby's movements.
We observe babies making breathing motions, which is essential for the development of their lungs. We see them sucking their thumbs, and indeed there are often babies that are born with calluses on their thumbs from sucking. This is obviously a reflex which is essential for life in the newborn period, but it's clearly hardwired in and present even during fetal development.
Another network present in the fetus is its circulatory system. From the first trimester, the growing child has circulated blood through its umbilical cord to the placenta, an elaborate system of blood vessels that grows inside the mother's uterus. Here, the fetus gathers nutrients and releases waste. But just as this lifeline delivers essential chemicals, it can transport harmful ones as well.
And many, but not all, compounds are either passively or actively transported across the placenta. It used to be thought that it was a complete filter against all potentially hazardous compounds in the maternal circulation, but of course we know better than that now.
Some chemicals that cross the placenta interfere with development. Others, such as those found in cigarette smoke, disrupt the ability of the placenta to provide nourishment.
Tobacco, as you know, causes a constriction of blood vessels, and this is quite pronounced in the blood vessels that lead to the uterus. This causes basically a starvation diet to the developing fetus and results in a syndrome we call intrauterine growth retardation, where the babies are born in a small for gestational age.
The best way for an expectant mother to protect her growing child is to take care of herself with a proper diet and adequate exercise.
Fortunately for the fetus, they're able to mobilize the mother's iron stores, calcium stores, and protein stores for their own development. So much of what we talk about in terms of adequate nutrition, exercise, and so forth during the pregnancy is directed at maintaining and enhancing the health of the mother, since the fetus pretty well can take care of itself unless it's already in a compromised position.
Count his heartbeats there as well as
In guiding her patients toward good health, Dr. Grau shares in the wonder of bringing new lives into the world.
And the baby now has its legs crossed.
You're allowed to share in this very intimate and very joyous event in people's lives. And there is a bond that develops between women and their obstetricians. It's a very powerful and very moving part of people's lives.
She breathes in, she breathes out.
The miracle of reproduction unfolds in a precisely measured sequence of events. From a single fertilized egg, nature assembles structures of staggering complexity. From this marvel of engineering emerges a living being who may someday have a child of his or her own and take part in this ongoing cycle of life.